God, hello. You've changed a bit. Is your hair? To be fair, Mum, it is your hair. Finally, after months of waiting, here we are, finally! Benito's Explanations has returned! And what are we going to be doing to celebrate this marvellous occasion? We're going to be studying DNA cloning in E. coli. What? Yeah, I'm afraid so. I may be back, but unfortunately, the reason why I haven't been doing any videos is because I've been at university. So then, I had this brainwave, why not put two and two together? And end up with a brilliant result. Just for you. How exciting. And this is it. DNA cloning in E. coli. But don't worry, I'm not going to stay like this completely. Hopefully soon, and I do promise soon, there will be some more amusing stuff for you to watch. I can declare officially that there will be an Into the West Christmas special for a start, so that's something to look forward to. But who knows what will go on between here and then. But for now, we're going to concentrate on this. Now, just like as I've said in my previous revision videos, I do not claim to be an expert on this whatsoever. In fact, I only learned this stuff a couple of weeks ago, so I'm nowhere near an expert on this. This is just my way of helping me to revise, and hopefully, with a bit of luck, it'll help you as well. You never know, that might just happen in this video. If not, tune in anyway, you know, it'll give me a view. We're going to be covering some relatively basic stuff in comparison to the other stuff I'm doing in this module. But obviously I'm not going to go down into the core basics of genetics here, because otherwise I'd be here all day. So if you do not know any genetics at all, why the hell are you watching this video in the first place to start off? Secondly, maybe go back to see some other videos if you are dying to know how cloning in E. coli is done. You know, you might just be doing that. You might just be sitting in your armchair and thinking, how does that work? Fair play to you. So let's give it a go, shall we? This module is basically about how scientists can mess around with DNA, can move about, take bits out, add things onto things, all that kind of stuff, all for the greater good of society, we think. To the everyday person, when someone hears the word clone, they'll probably think that's something you only see in Doctor Who or whatever. But no, it's happening here in biology, and in fact it's happening all the time in the natural world, not just in the laboratory. Basically, if you take any organism and propagate its genetic material without causing any change to it whatsoever, that is basically a clone. Now, we're going to be talking about cloning in E. coli just because that's a sort of standard organism that biologists use. It's quite easy to manipulate, in particular the K12 strain of E. coli, which is slightly more safe to use in the laboratory. So why are we cloning in E. coli? Well, usually it's because we want to introduce a particular gene or section of DNA to that E. coli. So that E. coli cell will multiply, it will propagate and produce more and more of that gene. And with more of that gene, that means it will produce more and more protein, and it's that protein that we most likely want. But how do we get a piece of DNA, which is from another organism, it could be from anywhere at all, so the DNA has been extracted, how do we get that into E. coli, and how do we get that E. coli to make the protein that we want? Well, we can't just take a bunch of E. coli and chuck a load of DNA at it. That would not work, unfortunately, although it would be great if it did, wouldn't it? Probably not, actually. So instead, we use a vector. So a vector is a way of transferring that DNA, packaging it, and placing it into our E. coli cell. Now, there are loads and loads of different types of vectors, and hopefully in this video we're going to be going through probably the three main types. The first one, which I came across at A level most certainly, is the plasmid. Plasmid is a circular piece of DNA found in most prokaryotes. Now, DNA in prokaryotes is circular anyway, but these are apart from the main circular chromosome. These are little tiny, smaller, circular, double-stranded DNA. Now, usually they contain specific genes for things like antibiotic resistance. In this example here, we've used ampicillin as the antibiotic. They also have an origin of replication. An origin of replication is where replication starts. And this is a special piece. This is where we want to insert our new DNA. and it's called a polylinker. Or basically a multiple cloning site. What does that mean? A polylinker is where there are many restriction sites. What's a restriction site? A restriction site is a sequence of DNA in which we could introduce an enzyme, a restriction enzyme, in, and it will cut the DNA. If we cut the DNA, that means we can insert the piece that we want right in the middle. Now this particular restriction enzyme will cut here and here at exactly the same points. Obviously we mustn't forget that the DNA molecule continues going down that way. Now these bases now have nothing to base pair onto. So that's why we call them sticky ends. Because if there was a sequence, another sticky end, which is complementary to this sequence here, the TGCA, it would quite happily stick to it, it would bind. So if we cut our DNA from whatever organism it is with the same restriction enzyme as what we're cutting our plasmid with, we'd end up with the same sticky ends. So that means when we introduce our DNA, they'll quite happily stick to these bases here and be joined on. Doesn't have to be sticky ends, you can also get blunt ends. So if we go back to what I showed you earlier, so we're cutting it straight down the middle. Now obviously if we cut it down straight down the middle, all the bases are still base paired. So there are no sticky ends in this case. This is what we call blunt ends. So that's basically how a restriction enzyme works. It goes in, cuts DNA, preferably produces sticky ends, and then you can mix your cut up plasmid with your DNA, cut up DNA, and stick them together and then ligate them. Ligation is done by the enzyme DNA ligase, and that is what connects up the phosphate sugar backbone, which is DNA. Now we have one problem here. If we cut the plasmid, say at this point, with a restriction enzyme, let's just give an example so you know the name of restriction enzyme, EcoR1. Okay, that's a restriction enzyme. Now, if we cut the DNA, as I said before, and produce sticky ends, then the two strands which have been cut, there's nothing really stopping them coming together and sticking back together again and not incorporating the DNA which we want to introduce. Now we don't want that to happen, so how do we stop that from happening? Well, what we do, we add an alkaline phosphatase, and this is a process known as dephosphorylation. Now we should know, hang on, I'm going to have to speed up again. I don't know whether you can see that there, hopefully you can. Now we know that DNA is made up of repeating nucleotides, and each nucleotide contains a phosphate group, a deoxyribose sugar, and a base. And each deoxyribose sugar bonds with the phosphate on the next nucleotide, forming a phosphodiester bond. Now, what dephosphorylation does is it gets rid of the phosphate group. That's what the alkaline phosphatase does. So if we get rid of the phosphate group here, then DNA ligase won't be able to fit the two strands which we've torn apart. It won't be able to fix them back together again. It won't be able to form the phosphodiester bond because there isn't a phosphate there to bond with. It would just be bonding with another sugar, which it can't do. So if we do that to our plasmid, it's very important that we don't do it to the DNA we want to introduce, because otherwise nothing will bond. That's not what we want. But if we do that to the plasmid and then introduce DNA, then the only thing that these sticky ends will bind to and that DNA ligase will ligate to will be the introduced DNA, so you won't get any religation of the plasmid. So, once you've done that, you've ligated the DNA that you want to put in your vector, you've got it in the plasmid. Everything's hunky-dory. Then what you're going to do is going to introduce this plasmid that has your recombinant DNA in, and you're going to introduce it into an E. coli cell. How are you going to do that? You normally suspend this plasmid mixture in the E. coli colony in calcium chloride, and then you add some sort of environmental shock. That could be heat shock, or it also could be probably the most efficient method, which is electroporation. That's adding an electric voltage. I don't think we need to go into the details of that, because quite frankly, I don't know. So through that method, the E. coli cell can absorb the plasmid. So the plasmid is in the E. coli cell now. It's ready to replicate. But obviously, in your colony, on your agar plate, you're going to have some E. coli cells which have taken up the plasmid, and some which haven't. So how do you distinguish between the two? Now we were talking about this antibiotic resistance gene earlier. Well, this is where it's really going to have its starring role, because this is how you're going to test for it. For that, I'm going to have a drink of water. So how does the antibiotic resistance gene come into the screening for seeing which E. coli cells have taken up the plasmid? Well, any E. coli cells which have taken up the plasmid will be resistant to antibiotics, in this case ampicillin. So if we were to add any antibiotics to the agar plate or our colony, any cells which don't have the antibiotic resistance gene will die. Sad times, I know. But the cells which have taken up the plasmid will be resistant to antibiotics, so will therefore survive, and that's the kind that we want. So hip hip hooray, that's how you screen for it. Okay, so that's how you tell whether the plasmid has been absorbed by the E. coli. But how do you tell whether the gene you want is actually in the plasmid at all? That's still an issue. And obviously that risk is very much reduced due to our dephosphorylation using alkaline phosphatase. But you still can't be sure. But in our example, specifically the PUC18 plasmid, the place where we're inserting our gene is in a specific place. It's in a particularly clever place. It's just in front of the promoter sequence for a gene called the LAC-Z gene. Okay, what is the LAC-Z gene? Oh, it's like change the light in there just to spice things up a bit, you know. So what is the LAC-Z gene? Well, the LAC-Z gene, or the LAC operon, is a sequence of DNA which codes for the protein beta-galactosidase. And beta-galactosidase is an enzyme which breaks down lactose. Now, why this is so useful in our scenario is that it reacts in a specific way to the chemical X-gal. Right, now what's X-gal? No, it's not the way a chav speaks to his ex-girlfriend. It is actually a chemical, and it's a very long chemical, so we just call it X-gal. When beta-galactosidase is mixed with X-gal, it turns the solution blue. If there is no beta-galactosidase, the solution stays white. So that's the starting point. Now, as I said, the E. coli has this LAC-Z gene on its main chromosome, not on the plasmid. Now, the wild-type strain of E. coli has this LAC-Z gene on its chromosome, but you can get mutants which don't produce the desired protein. For example, the mutant that we're going to talk about is the LAC-Z delta M15 mutant. Dangerous stuff. Beta-galactosidase is made up of two fragments. There's the alpha fragment, and then there's the omega fragment. Now, the problem with the LAC-Z delta M15 mutation is that it does not produce the alpha fragment. So, that means if we introduce X-gal into this solution, then we're just going to stay with a white solution because there's no beta-galactosidase being produced. However, if we insert a plasmid which has the coding region for the alpha fragment of beta-galactosidase, then the alpha fragment produced in the plasmid will combine with the omega fragment produced in the chromosome and give us complete beta-galactosidase. Now that's alpha complementation. So if we had a plasmid with the lac Z gene coding for the alpha fragment present, insert it into E. coli, then that E. coli expresses that alpha fragment, then we will get complete beta-galactosidase. Now how on earth does this relate to this sequence of DNA that we have included onto our plasmid? Well, if our polylinker, if our multiple cloning site is within this region which codes for the alpha fragment, we can directly screen to see whether the E. coli have taken on recombinant plasmids or non-recombinant plasmids just by looking at the effect when we add X-gal. So, remember, if there's beta-galactosidase in the solution, and we introduce X-gal, it's blue. If there isn't, it's white. So if we introduce our gene of interest and stick it right in the middle of this gene here, which codes for the alpha fragment of beta-galactosidase, then the production of the alpha fragment is inhibited because something's stuck in the middle of it. It can't work anymore. No alpha fragment can be produced. So only omega fragment is being produced. Now, the omega fragment can't work on its own. So if we inserted our gene and introduced X-gal, the solution would still stay white. And that will indicate to us that we have a recombinant on our hands. So that's basically it. If we've screened for the recombinants, we've screened for the E. coli cells which have taken up the plasmid, then we can be pretty sure that the gene that we want is in the mix and is being replicated inside the E. coli cell, which is very useful. So that'll keep on dividing and producing many clones. A collection of clones is known as a library. There you go, you know. The days when a library was a place where you got books, so last century. You might think, why not just stick with the plasmid? It seems perfectly okay. Why introduce another vector at all, you know? Well, the problem is, there's only a certain amount of DNA which you can reliably fit into a plasmid. To put in larger sections of DNA, you need another type of vector. So for the moment, we're going to have to say goodbye to the plasmid and hello to this guy. Yeah, not quite as beautiful as the plasmid, but that could just be my drawing, you know. Maybe I'm giving them a bad service. But this is the Lambda phage, the Lambda bacteriophage. Now this is something completely different, really. The Lambda phage is a bacteriophage, so that is a type of virus that infects bacteria. More specifically, the Lambda phage, it infects E. coli cells. Very good. So that makes it a very useful vector for introducing DNA into E. coli cells, because that's basically what a virus does. This virus is basically just composed of nucleic acids in the middle. It's double-stranded DNA, and that's encapsulated in this head piece here and this tail. The tail is sort of like the cork into the bottle, if you like. It screws everything in to this head here. What happens when a Lambda phage infects an E. coli cell? Right, let's find out what happens. That took a lot of effort. Right, what am I trying to demonstrate to you here? This is how a Lambda phage infects a bacterial cell. So let's start with this in the middle first. So here we have our bacteriophage. It'll inject this blue DNA. It's not really blue in real life, obviously, but you know, that's how I've colour coordinated it. This is the E. coli DNA here, that's the circular chromosome. Now there are two possible paths that could happen from here. The first one, the slightly more boring one if you like, is lysogeny. This phage DNA will be incorporated in the bacterial chromosome, and that bacteria will just carry on replicating, producing many copies with the phage DNA incorporated. What the biologist wants is a bit more drama in the mix. So what we prefer is the lytic reaction, which basically causes the E. coli cell to burst when the phage enters, and that causes the phage to replicate, producing many more phages. So this is a lytic reaction. Lytic obviously means splitting, and that is basically what we're doing. So that produces more bacteriophages. We could go on and infect loads more E. coli cells, and the whole thing can happen again. So we will have tons and tons of bacteriophages by the end of it, which is what we want, especially if it includes the DNA which we've inserted into it. And this lytic reaction carries on happening on your, let's say, your agar plates. What you get is clear patches. They're called plaques. And that's where there's a high density of phage particles being produced, because there's the clearness is of it because of the lack of E. coli cells, because they've all been killed. So that's called a plaque on your lawn of E. coli cells. Now, before we go into talking about how we introduce DNA into bacteriophages' DNA, we have to know a little bit more about the mechanisms of replication of the bacteriophage. Now, the DNA inside a bacteriophage is linear, although on each end it has a 12 base 5' overhang. So it has 12 bases which aren't paired to anything. It doesn't have a complementary base, so they're single-stranded at the end. And these 5' overhangs can join together to form a circle. It's a nicked circle because the phosphate sugar backbone isn't connected by DNA ligase, but it forms a circular molecule. And that's essential to how the bacteriophage DNA replicates. It's a type of replication known as rolling circle replication. What is rolling circle replication? This genome within this circular DNA, it'll keep on going. Okay? So let's say the origin of replication is here, where I've started my arrow. All the enzymes involved in DNA replication, like DNA polymerase, let's just say, DNA polymerase will carry on going all the way along here, replicating all the DNA all along, and when it gets to here, it will carry on going around again. So what you end up with, your daughter strands, are just repeated copies of the same genome. It's called a concatemer. The length of each genome is between what we call cos sites. So this is a cos site here, and this is a cos site here, that's a cos site here. This concatemer is shoved into the phage head, and when the correct length of DNA is in, an enzyme will cut, the tail will come in, and there you have a brand new bacteriophage, all done and dusted, ready to be packaged off and infect E. coli cells. Cheers. Now, the amount of DNA that can fit into a phage particle is about 51 kilobases. So if the gene or piece of DNA that you want to insert into your Lambda genome, once you've added it, is greater than 51 kilobases, then you're in some trouble, because that means it just won't be packaged. The heads won't form, the tails won't form, you won't get a Lambda particle. Actually, there are two types of Lambda vectors. There are insertion and replacement vectors. Now, they basically just speak for themselves, really. Insertion vectors is when you just add something in to the Lambda phage, into the Lambda phage DNA. The maximum length of DNA which you can add is 14 kilobases. Why? Because there's also a minimum amount of DNA which the phage will allow to package, and that's about 37, 38. If you have a 37 kilobase Lambda genome, then you can have a maximum of 14 kilobases to it. It's obviously likely to be much less than that, but that's the maximum you can have. So this is a simple example of a Lambda insertion vector. This is the Lambda GT10 vector. Now, one of the problems with an insertion vector is that if you're cutting the Lambda genome with a restriction enzyme, there could be loads of restriction sites for that restriction enzyme anywhere along the genome. So before you do this, you're probably going to have to modify or delete them or treat them in some way. So when we add our restriction enzyme, it doesn't cut open that section of the DNA as well. Because otherwise you'd have loads of bits and pieces of all sorts of rubbish going on in your mix. This bit that I've shaded here is called the immunity region. You don't really need to worry about what it is, but it's a gene basically which is functional in the Lambda genome. If we add EcoR1 and insert a new piece of DNA, that gene will not work anymore. And as you can see, once this is cut, we can split the DNA into the left section and the right section, the left arm and the right arm. So the other type of Lambda vector is a replacement vector. So that means you've got a piece of DNA on your Lambda genome known as a stuffer fragment, and you replace that with the fragment that you want. So really the overall size of the DNA hasn't really changed that much. It might do a little bit by a few bases, but you're just replacing one DNA sequence with another, with the one you want. So unlike in the insertion vector where we're splitting it into two, this time we're splitting it into three, aren't we? Now how do we distinguish between those three pieces? Well, we put them on an agarose gel, conduct gel electrophoresis, and sort them out by their size so we have the right fragment in our bag and we can discard this stuffer. By doing this, we're significantly increasing the cloning capacity of the Lambda vector because we can put more stuff in because we're taking stuff out. We can't take too much stuff out, however, because that will affect the overall stability of the Lambda genome if it's got too much stuff which it's not really that used to. And now it's time to move on to our last vector of the episode, and oh, there it is, all the chance of that. Now, you might be thinking that, hang on, this looks familiar. That looks like a plasmid, right? This is a Cosmid, and it's basically the in-between. It's a little bit plasmid, and it's a little bit Lambda phage. It's a hybrid, basically. Now, what's plasmid about it? Well, it's a circular piece of DNA, just like a plasmid. It also has genes for antibiotic resistance, just like a plasmid. It also has cos sites, just like a lambda phage. It replicates by rolling circle replication. Now, the actual size of the Cosmid is actually much smaller than the plasmid. It's probably no more than about 5 kilobases. So, but that means you can fit a lot more DNA, even more than a lambda phage, into a Cosmid. So that means you have a much larger cloning capacity once you insert your vector at a certain polylinker using a restriction enzyme. Once that's happened and replication happens, packaging takes place just as in a lambda vector with a protein coat, and it can be sent off and introduced to any cell, well, in particular E. coli, an E. coli cell you wish, and it will replicate in there. And in fact, packaging will only happen in the Cosmid if the actual length of the Cosmid is about 32 kilobases. So that means if you have a non-recombinant Cosmid, but it doesn't have any inserted DNA in it, then it can't be packaged, because I just said Cosmids are very, very small, only 5 kilobases. So that means there's a positive selection for recombinant Cosmids, which is really, really good. So that is basically it. Right, hopefully, I don't know whether I'm using that word correctly here, hopefully I'll come back with another video soon. So I hope you found that kind of informative. I mean, I'm not going to lie, I struggled a bit, but it was great fun, and I think it's really helped me with my revision. Hope it's helped yours. If you enjoyed it, please let me know. You know, it'll boost my ego up tremendously. And if you didn't enjoy it, also let me know, because it gives me points to improve on, what I can do better, what things I can improve on, basically. But if you did like it, go crazy, share it, you know, like for like, you know, I'll do all that sort of stuff, honestly. I'm desperate. But for the moment, see you next time.